Welcome to the summary video for the Introduction to Prokaryotic Cell lecture. This lecture is divided into two major parts. The first part of it is describing the basic structure, the basic anatomy of a prokaryotic cell. Remember, those are just bacteria. So what do bacteria look like, both on the basic structures internally and externally. And then the second half of the lecture lesson describes how to grow bacteria in the lab, and what they need to grow and be happy. So we'll begin with the classic kind of cartoon of a bacteria. Okay, so here is a kind of typical little bacteria, although that one's a little bit ugly, so let's try again. Okay, there we go, prettier bacteria. Okay, so bacteria, prokaryotic cells, are much, much simpler than eukaryotic cells when it comes to the structures that you find within and on top of them. The same basic chemistry is there in a broad sense, but there aren't all of these structures, the internal compartments that are holding special groupings of enzymes. Instead, it's all primarily just inside of the cell. So the insides of the cell, you will find fluid, the cytosol, referred to as the cytoplasm, the area inside of the cell. The fluid itself is called the cytosol. So all cells basically are water balloons. So the water part of the water balloon is universal, the cytoplasm. The balloon part, the outside part, is called the cell membrane, another universal thing found in all cells, eukaryotes or prokaryotes. Inside of the cell, you need the instructions to run the cell, the DNA, another universal thing. In bacteria, our double-stranded piece of DNA is hooked together in a big circle. So there's one piece of DNA as opposed to lots of pieces in eukaryotic cells. And the pieces are hooked together to make it a circle instead of linear pieces like you find in eukaryotic cells. And one of the major reasons for that is when you look at a piece of DNA, the most sensitive parts of it are the tips. Little Pac-Man looking enzymes can come along here and start nibbling away at the tips of your DNA. In a eukaryotic cell, that DNA is protected from those little Pac-Man guys by being inside of a nucleus. But in prokaryotic cells, you don't have a nucleus. So all of our little destructive enzymes, our little Pac-Mans, are sitting there next to our DNA and available. So what happens, instead of having a special container around the DNA, you make the linear pieces of the DNA hook together and make a big circle. So there is no end for the little destructive exoenzymes and little Pac-Man guys to come and eat. Okay? So you have one circular piece of DNA. That area within the cell where you find the DNA kind of coiled up is called the nucleoid. It is not a separate container. It is just an area within the cytoplasm where you find the DNA. So all cells have the fluid, all cells have the membrane, all cells have DNA, and all cells have ribosomes, the machines that take the instructions in the DNA and actually produce proteins, the products that you need. The ribosomes are the chef, right? If your DNA is the cookbook, ribosomes are the chef, and the protein would be the food at the end. Okay, so when you look inside of a prokaryotic cell, you don't have a lot of structures. You basically have your nucleoid where our DNA is. You have ribosomes floating around, and you have some other basic things, but primarily that's it. You do have some optional things that are only found in bacteria, such as plasmids, such as endospores, two things that I'll let you read about and watch videos about on your own. On the outside of bacterial cells, instead of just having the basic membrane, all cells have a membrane. Most bacteria have a layer on top of the membrane called the cell wall. Whoops. The width of the cell wall varies. If it's a gram-positive cell, then you have a nice thick cell wall. If it's gram-negative cell, you have a much thinner cell wall. But the cell wall is just a very, very thick sugar layer sitting on top of the cell membrane. The cell membrane is like a balloon. It's a pliable, stretchy, kind of slightly vulnerable layer. The cell wall would be like a sugar brick wall on top of it. Some bacteria have a layer on top of that cell wall, another thin, stretchy, balloony membrane. So they have two membranes and a wall and an inner membrane. Those would be our gram-negative bacteria. And you can read about that and see a much prettier picture in the videos. Okay, so there are potentially some additional structures that you find on the outside of the bacteria. All of these are optional things. So you could have flagella, which flagella are all about motion, motility, movement. You could have fimbriae, which flagella look like kind of like big tentacles sticking out. Fimbriae look like little hairs, and they are sticky, so they are basically like little Velcro, that allow the cells to stick to things. Another option that you may find on the outside of your bacteria are pili, or pilus for singular. And pili are a way to communicate between two cells. So pili are basically like big hollow tubes. And what happens is some cells can create these if they find a partner, a good match cell, called a recipient cell. And this original cell will create this pilus and will puncture a hole into the recipient cell. And then this pilus becomes like a tunnel for DNA to pass through from one cell to the other. So it becomes kind of a little DNA exchange tunnel. So this is basically how one way that bacteria can get new DNA. So it's a way of having sex, basically. Remember, the biological definition of sex is a cell that has different DNA afterwards. So in this case, you're not creating a new cell. It's taking the original cell that was there and mixing up the DNA. So the pilus is for DNA exchange, if a bacteria can produce it. And the final structure that some bacteria may have is an additional layer on top of the cell walls and cell membranes that are already there, an additional layer called a glycocalyx. It's all made of sugar. If the sugar is kind of sticky and wet, it's referred to as a slime layer type glycocalyx. If the sugar is hard and kind of dehydrated, it's referred to as a capsule type of glycocalyx. I always think of it as like the crunchy candy shell on the outside of the M&M would be like a capsule, and like a caramely, sticky layer would be like the slime layer. So you can watch the videos and read about the exact functions and advantages of having one versus the other. Again, all of those appendages, the flagella, the fimbriae, the pilus, the glycocalyx, those are all optional things. Okay. So the second half of this lesson is all about how to grow our bacteria. And we're primarily talking about how to grow them in the lab, but really what we're doing in the lab is trying to mimic what they'd like in nature so that we can grow them in a controlled way. So the things that are required, the conditions that are required to grow bacteria are very much the same as the conditions that are required for us to live. So you have to have the proper amount of food, you have to have the proper amount of water, you have to have the proper amount of oxygen, you have to have the proper temperatures, and whether you think about it or not, you have to have the proper pH. Go stick your hand in acid and see if the proper pH matters. So it's the same for us as it is for them. So the big thing with this part of the lecture is just a lot of vocabulary. For humans, we have to have oxygen or we'll die. And there are some bacteria that are that way too, but then there are some bacteria that absolutely cannot tolerate oxygen. And if oxygen's in their environment, they'll die. So you can't make the assumption that oxygen is good and required, not for bacteria. The same with pH. For us, we need neutral pH. That's what our cells like. But there are some bacteria that love very high very, very, very high pH, very basic. There's some bacteria that like really low pH and like to live in acid. In fact, there's a bacteria that can live in our stomach acid, which is about pH 2. Very, very acidic. So with bacteria, you can find examples of organisms that live in all kinds of weird ranges for these 5 basic environmental conditions. You can't just assume like you can with animals or plants. In fact, most of the other organisms. So with this lecture, you're going to have to learn the vocabulary word that represent the different ends of the spectrum that you find for all of these conditions. So water, how you get the proper amount of water is kind of more formally, scientifically referred to osmosis or osmotic balance, water balance. And this is, you may have bumped into the terms hypertonic, hypotonic, isotonic before. That's what this is talking about. In this class, I'm not going to test you on those terms, but I want you to have the basic idea and understanding that the solute levels like salts and sugars will influence whether water goes into a cell or out of a cell. And bacteria have the same response that we do. They have a cell wall on the outside of them, which gives them a little bit of protection from exploding when water comes rushing inside. But it doesn't help when they're dehydrating. The cell wall doesn't keep water in. It just keeps them from rupturing quite as quickly, but it can still rupture eventually. So food. Bacteria have the same basic food requirements. Carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus are the primary ones. And then there are trace elements like calcium and potassium, all of basically the same things that we have. Go look at the bottle of, the back of your multivitamin. They have the same needs basically. But how do we deliver food in the lab? Through media. So you've gotten a taste of it already in the lab most likely. And we will basically spend the entire portion of the semester working with different media. But media is the way that we deliver food to the bacteria, giving them all of the nutrients and salts and sugars and all of the stuff that they will need. So there are a few vocabulary words that go with media that you will need to know: selective media, differential media, things that we'll use a lot in the lab. So I want you to know those and be comfortable with those words so that when they come up in the lab, they're not confusing to you. Okay, so hopefully this part of the lecture will be, or this lecture in general will be pretty straightforward. There's several vocabulary words that you'll need to learn, kind of flashcard type things. But conceptually, hopefully, it will be pretty straightforward to you. But as always, if you bump into any questions, have any confused, feelings about any of the material, please post in the Muddiest Points discussion board so that your peers and I can help you through it. Good luck.